All right, a huge welcome to all of Mr. Cheese students and followers. So glad you're here. Today, we're going to tackle one of those exam questions on meiosis. You know the one? The kind that always seems to trip people up, but not anymore. Let's just dive right in and get this sorted.
Okay, let's be honest. Have you ever stared at one of those meiosis diagrams in an exam and your brain goes completely blank? Yeah, you are definitely not alone. So what we're going to do is break down a really classic, kind of tricky question, piece by piece. I promise by the time we're done, you're going to feel so much more confident facing these.
So this diagram right here, this is our puzzle. And believe it or not, all the answers for the first few marks are hiding in plain sight. It's all about knowing what to look for. So let's put on our detective hats and decode these visual clues together.
Okay, first things first. We've got to know who the players are in this game, right? The very first part of the question asks us to name structures A and B. And trust me, getting these right is the foundation. Mess this up and the rest gets shaky, so let's lock this in.
All right, let's get precise with the language here because that's where the marks are. Structure A, that's a homologous chromosome. Now listen up, this is a super important exam tip. The memo is really, really specific. If you write pair or bivalent, you will not get the mark. Seriously. So homologous chromosome it is, and for B, that's your spindle fiber or spindle thread. See? In biology, the exact words matter. A lot.
Okay, so we know the parts. Now for the big question. What phase are we even looking at? Well, if you look closely, you can see how those homologous chromosomes are lined up right down the middle, at the cell's equator. And that is the classic textbook sign of metaphase 1. Boom, nailed it.
All right, see how these questions build on each other? The next one, 2.3.3, asks what happened before this picture was taken. So we got to hop in our time machine and rewind a little bit. We need to figure out what went down in prophase 1 to get us to this exact point.
So prophase 1, what's the checklist? First, your chromosomes get all condensed and become visible. Think of them like wound-up balls of yarn. Then poof, the nuclear membrane disappears. Next up, those spindle fibers we identified earlier, they start to form. And then, the most important event of all, crossing over. That's the big one. And hey, for the exam, you don't even need all of these. Just a few will get you those 4 marks.
Okay, that last thing I mentioned, crossing over, it's such a huge deal that the exam dedicates a whole 5-mark question to it. 5 marks. The question wants us to explain how this tiny event inside a cell connects to, well, the biggest idea in all of biology, natural selection and evolution. This is where it all comes together.
So, what's the core idea the examiners are looking for? It's this. Crossing over is literally chromosomes swapping bits of their DNA. They're shuffling the genetic deck. And the result of that shuffle? You get gametes, sperm or egg cells, that are all genetically unique. And that simple fact is the launchpad for everything that follows. Okay, so how do we stretch that idea into 5 solid marks? It's a chain reaction. Step 1, a unique gamete leads to genetic variation in the kids. It means no 2 siblings are exactly alike, right? Step 2, some of that variation might be awesome for survival, a favorable trait. Some, not so much. Step 3, now imagine the environment changes. Suddenly, that one favorable trait makes a huge difference. The individuals who have it, they're more likely to survive, have their own kids, and pass on that winning gene. And that, my friends, is natural selection in a nutshell. That's your 5 marks, right there.
All right, we're on the home stretch now. The last part of this question is all about the numbers. First, we need to get the chromosome count right. And second, we're going to look at what happens when the counting goes horribly wrong.
Okay, here's a quick, easy mark for you. How many chromosomes are in a human gamete when meiosis is all said and done? The magic number is 23. You just have to remember that meiosis is all about reducing the number. It takes the full 46 chromosomes in a body cell and cuts it in half. Simple as that.
But, you know, biology isn't always perfect. The process can make mistakes. The next part of the question throws us a curveball. A gamete ends up with an extra chromosome 21. So, how on earth does that happen?
Well, there's a specific name for this kind of error, and you've got to know it. It's called non-disjunction. It's a fancy word that just means the chromosomes failed to separate properly during the anaphase stage. They got sticky. This can happen in either meiosis I or meiosis II. And that specific example, an extra chromosome 21, is what leads to Down syndrome. It really shows you how incredibly important it is for this process to work perfectly every single time. So, to bag those full 2 marks, you need a 3-part answer. Ready? Point 1, you have to use the word non-disjunction.
Point 2, you have to say that it was the chromosomes at position 21 that failed to separate. And point 3, you have to mention that this mistake happens during anaphase, either I or II. You hit those 3 points, and that's a guaranteed perfect answer.
And that's it. We just took apart a whole 15-mark question piece by piece. But look, the point here isn't just to memorize the answers for this question. It's about building a game plan, a strategy you can use for any meiosis question that comes your way.
So here's your cheat sheet, your master plan. If the question says identify, your eyes should immediately go to how the chromosomes are arranged. Are they in the middle? Are they pulling apart? If it says describe, think of it like telling a story in order, using action words like forms, disappears, occurs. And if it says explain, that's your cue to connect the dots to the big picture. Think genetic variation, natural selection, survival. That's what they're looking for.
I really hope walking through that was super helpful for you. Honestly, the key to getting good at this stuff is just practice, 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 and using awesome resources. And speaking of which, you should definitely keep this learning going with Mr. Chi online. He's everywhere: Instagram, TikTok, Facebook, YouTube, posting all sorts of amazing life sciences content. So if this explainer made things click for you, do Mr. Chi a massive favor. Hit that like button, share this with a friend who's also finding this tricky, and of course follow him on all his channels so you don't miss the next one.
So today, we totally nailed the how of meiosis. But every single process in biology also has a deep and fascinating why. So what's next? What other complex system are you itching to understand? Keep that curiosity going, never stop asking questions, and we'll catch you in the next explainer.